to my YouTube channel where we discuss the GCSE Biology AQA past papers and the resources to ace your biology exam. If you like my channel, please like, share and subscribe for more resources and materials. Here we will discuss one by one questions and break down so you could understand them better. So pause the video wherever you like. These are exactly the types of questions that show up in GCSE biology exams. Past papers are a great way to prepare for your upcoming exams. It's a window to learn about the paper patterns and trends in marking schemes. In this video, I'm walking you through topics addressing enzymes, plant tissues, osmosis, antibiotics, photosynthesis, cell division and cancer. Higher Tier 1H Year 2024 past paper. Not just the answers, but the exam technique that gets you full marks. Let's go. Let's begin our today's lesson with the question 3, which deals with enzymes and digestion. As we know, enzymes are biological catalysts speeding biochemical processes and catalyzing living chemistry. So let's read and start from first slide where the question part 3.4 of this past paper asks you to explain why the bread tasted sweet. Now the answer requires you to understand the table 2 data and the investigation of breads and its 3 marks question. So the answer is about the amylase. An enzyme is present in saliva. It breaks down starch in the bread into smaller, simpler sugars like maltose, and sugars taste sweet. Simple chain of logic. Now here's your top tip. When the question says explain, use linking words like because, which means that's how you connect the science to the observation. Let's move on to next part, question 3.5, which asks you why might the results not be valid, and the answer is the student may not have waited long enough for the reaction to occur, or there may have been contamination from other foods. One clear scientific reason equals 1 mark. Moving on to the next slide, and the next question is question 4, which deals with plant tissues and transport. Let's work out questions on next slide, on which question 4 asks you about the parts of plants and various plant tissues. Now if you read the part 4.1 of question 4, which asks complete the sentences. First part asks you the tissue with the most chloroplasts, and the answer is palisade mesophyll. Secondly, the tissue with many air spaces, spongy mesophyll. Thirdly, the tissue that differentiates throughout life, meristem. Let's move on to next question, part 4.2, that asks you to name material what strengthens xylem besides cellulose, and the answer is, and the best answer is lignin. Let me give you an example tip. The meshwork of fibrous network of cell wall is composed of strengthening materials such as cellulose, hemicellulose and lignin. Easy memory trick. Lignin equals line that keeps plant in line, upright. Let's read the next question, part 4.3, which asks you to process transporting dissolved sugars, and the answer is translocation. Next question requires your deeper understanding about active transport, and the AQA asks you command words such as explain the processes. So the question is 5 mark explanation. Part of the question here on slide is 4.55 marks here. Sugars move by active transport. Active transport requires energy. Energy is released during respiration. Mitochondria in companion cells provide ATP. Carrier proteins in the membrane pump sugars against a concentration gradient. Notice how each sentence adds a scientific detail. That's how you build 5 mark answers. 4.6 asks you to answer the change happening when undifferentiated cells become phloem cells. So examine requires your understanding of command word where he asks you to describe one structural change in phloem. Note it's a 1 mark question, hence instead of long explanation 1 fact would be enough. So the answer would be that end walls break down to form sieve plates, or the cell loses most of its organelles to allow more space for flow. Will award mark. Here is the exam top tip. Don't confuse. Transpiration is water in xylem. Translocation is the sugar in phloem. Remember, phloem transports food. Let's move on to the next slide where students investigate the effects of salt concentration solution on uncooked potatoes pieces. Hence the question 5 carried out osmosis investigation, not only the definition but also applying the understanding of the id scientifically. First question starts with the experiment setup. The part 5.1 of question, which is 6 mark method question. Step 1. Cut 6 potato cylinders to equal size. Step 2. Dry and record initial mass. Step 3. Add different salt concentrations to labeled tubes. Step 4. Place 1 cylinder in each tube. Step 5. Leave for a fixed time. Step 6. Remove Dry. Record final mass. Then calculate change and plot a graph. Top tip. For 6-mark methods always say, repeat the experiment. Calculate a mean. Control variables temperature, surface area, volume. That alone pushes you toward full marks. 5.2 Explain the result for the potato pieces in the 0.6 mol decimeter cube salt concentration. 3 marks. According to the graph in figure 4, at 0.6 mol decimeter cube the potato pieces show a negative mean change in mass approximately minus 0.2 g. Direction of movement. Water moved out of the potato cells and into the salt solution by osmosis. Concentration gradient. This occurred because the salt solution was more concentrated, lower water potential, than the inside of the potato cells. Mechanism. Water moved through the partially permeable membrane of the potato cells. 5.3 Explain why the result for the potato pieces at 1.0 mol decimeter cube was different from the result at 0.6 mol decimeter cube. 2 marks. Increased mass loss. The potato pieces lost more mass at 1.0 mol decimeter cube, approximately minus 0.3 g than at 0.6 mol decimeter cube. Reason. The 1.0 mol decimeter cube solution is more concentrated, creating a steeper concentration gradient, so water leaves the cells more rapidly or in larger quantities. Let's move to the next slide, which all about pathogens. The question 6 is about pathogens and antibiotics. As a scientist investigates the antibiotic resistance and you are asked to explain the results and as well as memory recall and name one antibiotic. Let's start with question number 6.1, which asks you to name one antibiotic. The answer is penicillin. Question part 6.2, why is the student wrong about disc C? Is a classical AQA command word where examiner check your analysis and interpretation of data while asking to explain the students was wrong using the figure 5. And the answer is there is a clear zone, a zone of inhibition, around disc C that shows bacteria have been killed or growth inhibited. Exam top tip. Bigger clear circle equals more effective antibiotic. Next question 6.3 asks you to suggest why concern over antibiotic resistance. Infections become harder to treat. Recovery takes longer. Mortality rates can increase. Next slide starts with viruses saying they cannot be treated with antibiotics. Now first part question 6.4 asks you the command word suggest why can't viruses grow on agar. Viruses need living host cells to replicate. Agar is just nutrient medium. Next question is part 6.5, which asks you why is it difficult to develop antiviral drugs. Viruses live inside human cells. Drugs may damage our own tissues while trying to kill the virus. Moving on to the next question, part 6.6 asks which disease caused by virus damaging white blood cells, and the answer is AIDS, and the name of the virus is human immunodeficiency virus, HIV. Let's move to the next slide where AQA examiner check your data analysis and scientific knowledge through investigation of photosynthesis factors on leaves such as starch test and limiting factors. Table 3 shows the details of each test. If you observe the statements and analyze them, followed by answer to question part 7.1, explain the results of 3 leaves. Leaf 1. No starch because black paper blocked light. Leaf 2. No starch because plastic blocked carbon dioxide. Leaf 3. Starch present because light, CO2 and water were available. Let's move on to next slide where student investigate another plant for 48 hours. Figure 6 shows the details of plant leaf. Now the let's answer part of question 7.2 where examiner asks you to analyze and answer the table 4, which is starch test. Results. Green part turns blue-black. White part stays orange-brown. Now next question, examiner requires your answer to be based on command word to analyze the answer of table 4 and explain the results. Question 7.3 asks you to explain the answer. That why is this answer? Green part contains chlorophyll. White part lacks chlorophyll. Let's move to the next slide, which begin with statement some leaves green parts become yellow due to ion deficiency. Let's answer the question 7.4 part about this ion deficiency, and the answer is magnesium. Moving on to the next question, part 7.5 where examiner check your deeper understanding of yellowing of leaves. What is this scientific term called as? And the answer is chlorosis. Next question, AQA examiners check your practical knowledge and understanding here. Question 0.76 part asks you to explain how can oxygen produced during photosynthesis and be used to measure the rate of photosynthesis. This is a very important concept and the answer I could be obtained count bubbles per minute or measure oxygen volume over time. Let's move to next slide where examiner check your data analysis and its interpretation skills. Here on slide the figure 7 shows a graph of limiting factors such as light, Carbon dioxide and temperature. The rate of photosynthesis shows on y-axis and light intensity on x-axis. On the basis of data showed on this graph, question 7.7 first asks you limiting factor definition. A factor in short supply that prevents the rate increasing further. Let's move on to the next slide, question part 7.8, which asks you to explain the increasing effects of temperature and carbon dioxide on rate of photosynthesis as shown in figure 7. The answer is by increasing CO2 increases rate of photosynthesis, and by increasing temperature increases rate, up to a point, because enzymes work faster. Moving on to the next question 7.9, which asks you to explain inverse square law. Light intensity is proportional to 1 over distance squared. Exam top tip. Chlorophyll is the pigment for autotrophic factory. No chlorophyll, no starch. Question 8 on next slide deals with cell division and cancer. Topic is mitosis and cancer. Let's begin with question number 8.1, where examiner asks you to before division, the cell grows and increases subcellular structures. Let's answer the question 8.2, name the structure Z, and the answer is cell membrane. Next question 0.83 part AQA examiner, check your mathematical skills where figure 8 is used for magnification calculation. The formula is shown here. Step 1. Calculate real length. Step 2. Convert units to match. Step 3. Divide image by actual. Final magnification. Approximately 8010 times. Exam top tip. Always convert units before dividing. That's where easy marks are lost. Let's move to the next slide, question part, where figure 9 shows the phases of mitotic cell division and one cancer drug X effect is shown to ask you on stage 1. Now the question 8.4 asks you to explain why cell cannot complete the cell division. Means if spindle fibers cannot attach, chromosomes cannot separate. The answer is straightforward, that no separation, no identical daughter nuclei. Based on figure 9 and drug X effect on stage 1, examiner asks you to explain in cell question 8.5. Why treat cancer by stopping division? The answer is cancer is uncontrolled cell division. Stop division. Stop tumor growth. 8.6. Preclinical testing. Testing drugs on live tissues in laboratories. Thank you for listening till the end. Please stay tuned for my upcoming content and like, share and subscribe.